Good morning, all. I am Purnima Verma, working as Associate Professor in the Department of Biochemistry, and I am here for rendering a lecture on nucleic acid based technologies. But before entering into my duty, let me first thank Kuhs and also Professor Dr. Annie Matthew for inviting me once again, to make a lecture on my DNA technology. Last time, when I was invited for making this session, I was quite impressed about what is to be done, from where I have to start, how can I pull the crowd towards me, how can I attract my audience towards my DNA, my PCR, my DNA profiling, etc. But of course, it went very well. But this time, of course, it's like blessing in disguise, because COVID has made such an impact on people that even a common layman can understand the term. Of course, they may not be knowing what is the DNA, what is going on in the body, how are the characters being transmitted from the parents to the offspring, of course, but they are now familiar with the term PCR. They know that in order to categorize them as COVID positive or COVID negative, they have to undergo a test named RT-PCR. Even though they may not be aware about the significance or relevance or what is being carried out, they know that it is supposed to be one of the gold standards or one of the confirmatory tests for diagnosing an infection. So it's, of course, COVID is having some positive impacts also, right? So I am going into my topic. I'm not extending my introduction. So let me first make my session. Have you seen this picture? What can you understand from this? This is nothing but our chromosomes. This is where our characters are being coded in some other forms, of course, in the form of 4 alphabets, namely A, T, G, and C, right? So they undergo some decoding process and give rise to some genotypic and phenotypic characters, making ourselves a parent's children, right? So my topic is nucleic acid based technologies. So we know that nucleic acids, they are certain biomolecules very much important for our life, and they belong to 2 categories, namely DNA and RNA. And in case of higher animals, it is nothing but the DNA, which is the genetic material that transmits the informations from the previous generations to the next generations, and they are responsible for bringing about all the characteristic features which make ourselves confirm that we are all born normal, without any genetic defects, without any inherited disorders, etc., without having any mutations, etc. So they have got a very important role in our body. And we know the importance and what are the functions, or what are the properties, what are the characteristic features of the nucleic acids and all. Right now, how can we apply these nucleic acids in our day-to-day life? That is being discussed in the topic named nucleic acid-based technologies. So have you ever seen the DNA coming out of our body? This is our DNA. This is how DNA appears when you are denaturing it and extracting it into a test tube. This is how our DNA looks like. So what is the relevance of this molecule? We call it as molecular biology, right? So the study which deals with the nucleic acids, the gene expression, the transcription, the translation, the mutations, etc., all of them coming under an umbrella, we call it as a molecular biology, which is indeed very interesting, very informative, very relevant nowadays. And these molecular diagnostic materials we can make use of as our tools to carry out certain manipulations, so that we can prevent the inheritance of certain disorders and all. So all of these coming under a branch of biology, we call it as biotechnology, we call it as molecular biology. So what is the importance of these molecular diagnostics methods, right? And what are the genetic techniques? How can we make use of in our day-to-day life or in our medicine, in the area of medicine? How can it be beneficial to the mankind? How can it be beneficial to the mankind? So all these are coming under the DNA based techniques or DNA based technologies or molecular diagnostic methods, right? So what is the importance? By manipulating certain genes, we can prevent the inheritance of certain disorders. We know that certain conditions, certain disorders are transmitted from generations to generations, right? So if there is any chance for the inheritance, we can prevent them. We can at least be aware of the inheritance of the disorders, and we can make certain precautions so that it is not carried away to the next generations. So all of these things can be done with the help of these molecular techniques or molecular biological tools, because certain cancers which are familial in nature, right, they are encoded by certain genes which are undergoing mutation. So these mutated genes can be isolated and can be corrected. And there are certain infectious diseases which can act as markers. We can identify them. So all these are coming under the applications of this molecular biology. So how can it be made possible? How can we do it in a clinical laboratory? Of course, it cannot be carried out in a primary or a secondary level laboratory. It is carried out in a highly sophisticated tertiary or quaternary level laboratories, because it makes use of certain micro techniques, right? It cannot be carried out in a careless manner. It needs some expertise. So it can be carried out in certain tertiary or a quaternary level laboratories by means of qualified personals, right? So, have you seen this picture? What can you see? A group of girls, beautiful girls. One among them is a tiny, cute little one. And actually, she is a baby or an infant or a girl studying in LKG or UKG? No. She is a normal girl having the same age of the other children. But she is of short stature, right? What makes her look like a dwarf, or why she belongs to the class of dwarfs? We all know that growth is influenced by a hormone known as growth hormone, which is synthesized from the pituitary gland, pituitary growth hormone, right? So the pituitary growth hormone is responsible for making us to grow in a normal fashion. If the growth hormone is secreted in an exaggerated manner, it results in gigantism. And if it is in a sub-level or it is in a subnormal manner, it gives rise to a condition known as pituitary dwarfism. Can it be corrected? Can this condition be corrected? Of course. Now it is able to correct. There are treatments going on those children who are having a reduced level of growth hormone secretion. How can it be corrected? We can artificially administer them with growth hormone. So how can it be done? This is made use of with our molecular biological techniques, right? So first what you have to do, you know that the defect lies in the growth hormone. So you have to first extract it, right? Growth hormone, it's a biological substance. It's a chemical messenger. We all know that it is secreted from which organ, it is getting acted on which all target organs and all. But we are not aware how it functions, or how can it be, if there is any defect lies in the secretion, how can it be corrected? How can you extract it out, or how can you make a girl or a boy who are having a tendency to show the dwarfism, how can it be corrected artificially? It can be corrected with the help of, we call it as recombinant DNA technology. The condition, the term we use as recombinant DNA technology or rDNA technology. It's indeed a very interesting field. It is, researchers are working in the various aspects of these chemical molecules, so that the chances for developing the inherited disorders can be reduced. So the pituitary growth hormone is found to be the second artificially synthesized product by the pharmaceutical companies to be implemented in the humans. And the first being, of course, our human insulin. So the human insulin and the pituitary hormone was the first two compounds or chemical molecules or biomolecules to be produced artificially by means of rDNA technology, recombinant DNA technology. But as as you think, it is not that easy. It's a cumbersome process. It's not that easy to isolate it and it can be administered in an individual. It's not that easy. It requires some technical processes. It has to require some technical expertise to artificially synthesize these hormones in the same condition as that is in vivo, because in vitro we are preparing these hormones. We are producing, or we are preparing it in the laboratories in an artificial way, but by providing the same conditions as in the biological system. Right. So that is the importance of rDNA technology. And how can you develop the human growth hormone or human insulin in artificial conditions? By means of a system or a condition, or by means of a technique we call it as cloning. Of course, we might have heard about the term cloning, right? The sheep and lamb was produced by means of cloning, but that cloning, it is known as, we call it as the reproductive cloning. Reproductive cloning is entirely different from the gene cloning, right? Gene cloning means, what do you mean by cloning? Cloning is nothing but the artificial production in an amplified manner in external conditions or in vitro methods, right? By making use of in vitro techniques. So that we call it as cloning. This cloning can be can fall into three different categories, namely gene cloning, reproductive cloning, and therapeutic cloning. There are three different types of cloning based on their functions or based on the applications. They are divided into three different categories, namely gene cloning, reproductive cloning, and therapeutic cloning. But as far as we are concerned, I'm talking about only the gene cloning methods, because that reproductive cloning is not it's not an accepted one. It's not a widely accepted one. It's follows certain ethical issues. So, but the gene cloning is not having any such technical difficulties. We can carry out in a laboratory, because we are doing nothing. We are just correcting a mutated gene. That's all we are doing. So gene cloning is it's going on in the laboratories nowadays. It's not at all an unethical one. It is purely an ethical one. So it follows all the ethical procedures also, right? So the gene cloning techniques. So this process in which we are artificially synthesizing a gene and administering into a patient, this phenomenon we call it as gene cloning. But again, gene cloning procedure, it follows numerous steps, right? Because the gene which is to be corrected has to be first identified. How many number of genes are present in a human body? Have you got any idea? How many number of nucleotide pairs are there in our body? We know that all of these characteristic features, it's all coded in the form of 4 letters: A, T, G, and C. Adenine, guanine, thymine, and cytosine. Right? These A, T, G, C combines in different permutations and combinations to give rise to the numerous types of proteins in our body, which are responsible for carrying out all the protein metabolic activities in the body. Right? So all these letters together forms around in an adult human around 3 into 10 raised to 9 number of nucleotide pairs are present. 3 into 10 raised to 9 nucleotide pairs. Such always they exist in the form of pairs. You know that DNA is a double-stranded helix, right? So they exist in the form of pairs, and such 3 into 10 raised to 9 number of pairs are present in a whole organism, in an adult human. That is, all these are forming, they are condensed in the form of chromosomes, right? And we are having a single cell is having 46 number of chromosomes, right? And a single cell contains how many DNA content are present in a single cell? A single cell contains around 6 feet of DNA. That's the total length of the DNA in a single cell, 6 feet. And we know that an average human contains around 3 trillion number of cells. So if you stretch the DNA, the whole DNA, it makes twice the length of the solar system. That is the length of DNA.